Next, we have an ENIGMA talk from Douglas Pike. Doug is a graduate student in my lab and part of the Rutgers Quantitative Biology graduate program. He's going to be defending his doctoral thesis next week, where he's been developing computational tools for protein design. He's a really creative and productive scientist, someone who really enjoys the fun of doing science, and it's been a pleasure working with him, for this time. He's been very productive. He's authored or co-authored over 15 articles while he's been in our group. And today he's going to talk to us about one project that he's working on coming up with a one-dimensional projection of protein structure that would make structure amenable to phylogenetic study. And Doug, it looks like you got your screen up.
Yep.
Take it away.
Can everyone hear me?
Yes. Yes.
You can see the slide.
Mm-hmm.
Okay. Really good talk so far. Interesting seminar overall. Thank you everyone for who spoke and commented. So the goal here is to try and dig a little deeper into protein structure, such that we might be able to predict evolutionary ancestry at a time deeper than are possible with amino acids due to basically the varying mutation over time that breaks down the similarity that's predictable from a sequence comparison in amino acids. This is nitrogenase, nitrogen reducing enzyme capable of converting N2 into reduced forms of nitrogen, such as ammonia. And as you can see, and other talks have reported, the structure of natural proteins is extremely complex. And you can see some patterns here. You can see the alpha helices, which are spirals. I think Paul calls them fusilli pastas, and beta strands are the light blue straight arrow looking cartoons, which are kind of like fettuccine. And then you have the spaghetti, which is yellow, which is kind of like an undetermined structure, an undefined structure. And then at the top, you can see the amino acid sequence, and I've colored the amino acid sequence by these different structural representations. And most of protein structure is in fact a combination of alpha helices, beta sheets, and then the other category of loop. And I'm going to go into a methodology that tries to, in particular, define what loops actually are. So very quickly, I'm just going to show you briefly the way that you quantitatively define a protein structure. How do you get these helices and strands quantitatively? You can look at two angles that go across the peptide bond of an amino acid chain called phi and psi, and you can plot the two angles together on a two-dimensional plot of phi and psi, and then you can see there's different regions where you have the green alpha helix, the blue beta strand, and then yellow is loop, which is it's not really a defined structure. It's some combination of structure. And then if you look at this plot a little more closely, you can see that there's a -1 slope sort of hidden in the trend of the occupancy of structure in proteins. This is a plot of frequency of the protein structure database called the PDB. So in the frequency plot, you can see sort of like a -1 slope trend. And that's because when you do principal component analysis of the database, you get about 2/3 of the variance in phi-psi distributions is an inversely dependent relationship between phi and psi. And that's why you get that sort of trending down of those different regions of Ramachandran space. And And this is primarily because residues per turn, which is a geometric parameter that I'll explain, it determines the hydrogen bonding patterns of protein structure, and it has a inversely proportional relationship between phi and psi, as I've plotted here in the geometric phi and psi plot. I've colored it by residues per turn, positive and negative, and I'll give you an idea of what residues per turn are. So a beta sheet is 2 residues per turn, and you can see on the left here, I have a very long peptide. This is actually only 30 amino acids, but I've made them all 2 residues per turn, like a beta sheet is. And you can see that that peptide chain is stretched out. That 30 amino acids is stretched out. It's like if you took a spring and you just pulled it all the way out. The diameter goes down and the length goes up. And you can see that the different hydrogen bonding oxygens and nitrogens in blue there are pointing in opposite directions, and that gives you the beta sheet hydrogen bonding pattern. And then at the other ones, the 3 residues per turn and 3.6, you actually have the same number of amino acids, but now you've crunched the spring down to a sort of a medium size, and 30 amino acids now is in a much shorter span of space, and there's more diameter in the helical turn, and 3.6 is an alpha helix. And then if you go all the way out to 5, you actually squish the spring completely flat and you get 5 residues per turn. And you make what looks kind of like a porphyrin, which is kind of cool, like a heme or a chlorophyll. And maybe there's some geometric relationship between short peptides with a residue per turn of 5 and the evolution of porphyrins. And then if you look on the right here, this is a loop in nitrogenase. You can see that I've colored them by their residue per turn number. And you can see that a loop is actually a combination of these geometries. It's not, unlike on the left, which is a repeating pattern of the same geometry and a very repetitive and symmetrical hydrogen bonding network, which is easy to define. This is some sort of meandering combination of different geometries going through the backbone. And some loops are flexible and they're not well defined in experimental structural characterization. Others are not. And in particular, it's those that, there's useful data to be explored in protein structure analysis. So if you bin the Ramachandran space by residues per turn, which is the plot on the left here, this is a 0.7 increment bin of residues per turn in Ramachandran space. You can sort of define structure beyond 3 groups, helix, sheet, and loop, and you can define them in actually 20 bins as a minimum, because the residue per turn difference between the smallest difference between secondary structures is about 0.6. So you need to go a little above that. On the right side of Ramachandran space, everything is on the phi greater than 0. Everything is a mirror image of phi less than 0. And at this point of phi equal to 0, this is why you can see the 2 different colors on each side. At this point of phi equals 0, the basically how pulled apart the chain is, like I was showing on the previous slide, stays the same, but the backbone plane is inverting around, and you're basically turning inside out across the phi equals 0. And then you have a mirror image on the right side of the Ramachandran space compared to the left. So anyway, with this binning, and if you apply this binning to the protein database, you can actually reasonably predict thermostability of alpha helix and beta that's been experimentally determined, where they make a point mutation in each of the secondary structures, calculate using mostly circular dichoism. I think most of the experiments are done in, determine the thermostability of the fold in the presence of different mutations, and you can estimate like what energetic cost experimentally there is for putting an amino acid in a given secondary structure. And by binning structure in this way in the PDB, we can actually predict this pretty reasonably for alpha and beta. The other geometric categories here have not been experimentally characterized, so we can't test those. But if you assume that alpha and beta being predictable is enough to use the rest of the data set, then you sort of end up with this statistical potential of structural binning and amino acids. And you can sort of use this as a Rosetta Stone for translation from amino acid to structure and from structure to amino acid. And then in proteins, this is what this looks like. So as I showed earlier, on the top, you have nitrogenase with 3 structures, loop, beta, and alpha, and you can see the sequence at the top. And then when you apply this structural sequence methodology using these bins and coloring the structure by those bins, you can see that the sequence has a lot more diversity. The colorization has a lot more diversity. The structure has more diversity because we're defining geometrically this sort of undefined space of loop. And many of these loops are rather rigid and actually hydrogen bonding. It's just not hydrogen bonding in a simple symmetric way, so it hasn't been well characterized. And so if you apply this method to compare 2 completely different proteins, say hydrogenase and nitrogenase from 2 completely different organisms, you may have a hypothesis that they're ancestrally related, but there's no amino acid evidence for that. If you try to 3-dimensionally structurally align these 2 proteins, the alignment software, some of the best algorithms, will align about 48% of the residues, but it'll calculate a 32 angstrom RMSD, root mean square deviation between the 2 structures, which basically is nothing. There's essentially 0 alignment. Its best was 48%, really bad. And then if you use our binning and do a sequence alignment using Clustal, you can capture on average around 40% structural sequence identity between these 2 proteins. And if you do that same analysis with amino acids, it's less than 20%. So I don't know if these 2 proteins are actually related, but at least we can quantitatively compare the structural relationship between these that's not sort of a binary of it can align 3-dimensionally or it can't. Using the binning to create a structural sequence, we can include insertions and deletions in the alignment like amino acids. We can also build substitution matrices and hopefully do evolutionary analysis of protein structure. So future directions of this method that we're currently working on, me and Saroj, primarily postdoc in the ENIGMA group, are working on this. We've, as recommended by Jana, we've generated a substitution matrix of the structural sequence, which is essentially just the geometric binning turned into letters. And our goal is to create a structural tree of life and compare it to the amino acid tree of life. And it'll be very interesting to see what happens there. My hope is that what we see is that there are structural relationships in proteins that are persistent beyond the range of sequence entropy over time. And one might be able to merge amino acid branches with structure. And then if we have a structural tree of life, we can also do ancestral sequence reconstruction calculations and try to build estimated models of the earliest protein structures. And I'd like to thank Vic and Paul for all their awesome guidance and support over the years, and many members of the Nanda and Falkowski lab over the years. And yes, I'm defending my PhD next week. Hopefully it goes good. Thank you to everyone for everything you've done for me over the years. It's been absolutely great.
All right, thanks, Doug. I think we can save questions for the session at the end, and we can move on with the program. So thanks to all the speakers so far.